Welcome to the 49th lecture of our course, and in this particular lecture we are going to talk about the carbon capture, sequestration and utilization processes and the role of environmental biotechnology and scope of environmental biotechnology in that. So, these are the following concepts which will be covered. We will briefly discuss about the problem of rising CO2 concentration and then CO2 sequestration, and the carbon sequestration processes, particularly the biological one, will be highlighted, and strategies for improving the microbial CO2 sequestration will be discussed in details, and followed by these the carbon capture and utilization processes will be highlighted. Now, elevated carbon dioxide is a partial cause of the complex phenomenon of climate change, and these elevated CO2 concentration is urging all of us towards the development of CO2 sequestration techniques to capture the CO2 from the atmosphere or to reduce its release. Now, the problems with the rising carbon dioxide is multidimensional. It is considered to be one of the most important greenhouse gas. It absorbs less heat per molecule than the greenhouse gases methane or nitrous oxide, but it is more abundant and stays in the atmosphere much longer. Increases in atmospheric carbon dioxide are considered to be responsible for about 2/3 of the total energy imbalance that is causing the earth's temperature to rise. And essentially, if you look at the data that is available for from the NOAA's annual greenhouse gas index, the combined heating influence of all major greenhouse gases can be seen, and we can identify that the role of carbon dioxide is pivotal. Now, the emission reduction and CO2 capture, sequestration and utilization is considered to be one of the priority areas of environmental biotechnology. Though physical and chemical methods are also developed and in practice for mitigating this high carbon dioxide concentration present in the atmosphere. Now, the emission reductions, where mostly the physical and chemical technologies are more prominent, it is found that these emission reduction will not be sufficient for limiting the global warming to 1.5 degree centigrade, a threshold which is considered and for a major ecological tipping point that is predicted. In combination with reducing emissions from the major industries who are releasing this carbon dioxide as part of their industrial processes, the climate crisis emerged due to this rising carbon dioxide concentration must also be addressed by active carbon capture, sequestration and its utilization via the cooperation between nations and industries in order to reach out to a net zero sustainability goal which is set for the year 2050. Now, how to manage this enhanced CO2? So, one idea is to reduce the emission that we talked about, that the industries are trying to develop technologies to reduce the emission, but at the other hand we have the prospects or we have the opportunities to use technologies or develop technologies, particularly using the microorganisms that to sequester or remove the atmospheric carbon dioxide and convert into a form which is safe and remain in the sequester form for a long period of time. And more to that, if we can convert that carbon dioxide through biological systems, microorganisms in particular, into valuable products. So, carbon sequestration basically can be done either by biological or by physical approach or non-biological approaches, and also using different tools and methodologies of engineering concepts. Now, new innovation and technology which are being developed has enabled the possibility of carbon capture and sequestration, which is conventionally abbreviated as CCS, with high investment cost and is akin to waste management. So, that is considered to be one of the drawbacks of the non-biological processes, because investment cost and also the waste management, waste which are likely to be generated from that. So, more interest has been focused on CCSU biotechnology. Now, U comes because of the utilization part. So, carbon capture and sequestration CCS, and then utilization. So, it is not only sequestering the carbon, but also converting the sequestered carbon into some form which is useful for the humankind or for our industrial or other purposes. So, there biotechnology plays a big role, and that actually is considered to be creating a value chain from the carbon which is being captured. Now, how to get these new CCU technologies? Now, an important part of the solution will be realized by innovative biotechnologies that recapitulate and tap the large scale ecosystem that underpin major global carbon cycle. Now, it is important to understand that these large scale ecosystems like the soil, like the ocean, like the deep earth crust, they have been cycling carbon for billion years. From the very beginning of this earth or the planet, they are cycling, or they are the house, or they are the place where the carbon is being constantly being recycled from different solid phase or rock hosted carbon to the atmospheric carbon. So, it is important that biotechnologists, particularly the environmental biotechnologists, should try to look back and see how these large scale ecosystems are functioning and what we can learn from them, the global carbon cycles, and then implement those in developing the CCSU. The innovation process will also be expedited by translating the fundamental knowledge we already possess, because we have lots of knowledge, information about the carbon cycling which are taking place in these large ecosystems, and we need to actually translate this knowledge into technology that store carbon within the earth large biomes or deliver to new value chain. Like for example, soil carbon cycle is known, pretty well known, but if we want to develop a technology, we need to look into the soil carbon cycle into that aspect, so that the carbon which is naturally sequestered within the soil system can be developed as a process to mitigate this high carbon dioxide into the atmosphere. And not only that, we need to also develop technology from our knowledge base, so that the sequestered carbon maybe from soil itself is converted to some kind of valuable products. For that, we need to harness the unique biological functions. Now, how will this be done? Now, we need to combine the contemporary ideas from the ecology and biotechnology, which are basically the core of the environmental biotechnology and which are associated with three major biospheres: plant soil systems, deep subsurface, and marine microbial ecosystem. So, these three systems are identified because these are representing the largest ecosystems where natural carbon cycles are going on, and so far we have partly understood, at least with respect to some of these systems like a plant soil system for example, to some extent the marine microbial ecosystem. Deep subsurface however is not very well understood by the scientists, but once we identify that can be a large repository of the global carbon, increased attention has been paid in last 2 decades at least to understand the deep subsurface carbon cycling processes. Now, there is a 3-step process identified to guide the cooperation between the ecologist and the biotechnologist, more specifically for the environmental biotechnologist. So, we will see what are these 3-step processes. These are called ecosystem inspired biotechnologies, and these have inevitable roles to reduce the accumulation of CO2 and methane in the atmosphere. Now, what are these steps? The first step is to identify the accessible control points or for the CO2 and or methane cycling, because we know that within the soil system this carbon and methane are being cycled, rather the carbon is cycled through this carbon dioxide and methane. Now, what are the accessible control point? What are the junctions between the engineered processes and ecosystem components? And that requires possibly the fundamental and applied scientists to work jointly and recognize what is already understood and which knowledge gaps need to be filled in to effectively render the ecological insights into new or enhanced engineering design principles. So, that is something which will help us to translate the knowledge that we have about the carbon cycling into an effective technology. Now, the scientific knowledge of carbon cycling and engineering solution for CCSU are not in themselves enough to spur the action. Knowledge is not enough, we have to actually get the knowledge gaps identified and work together, so that we can fill those gaps and then develop the technologies and translate the knowledge into effective technologies. And this also requires the third step is fostering the awareness of the problem and close the gap between the fundamental research and industrial action. So, it has been clearly identified in a very recent paper in One Earth. So, the reference is cited here is all, and also we have placed the reference at the back of the end of the presentation, that 7 main sinks are there within the global carbon cycle. So, this has been known for decades, but if we want to look very critically, that the 7 sinks are very well delineated, like in the atmosphere, in the plant, in the form of the fossil fuels, in the soil, in earth crust, in deep ocean and in the surface ocean. So, these are the 7 sinks identified. Now, one goal of ecosystem inspired CCSUs related biotechnologies is to find the control points in order to for the both natural and human influenced fluxes. So, how natural carbon flux like the yellow arrows are indicating the carbon movement, how the anthropogenic activities and how the natural activities are responsible for controlling the carbon fluxes within the 7 sink, and that possibly will be important to identify the points that pull more carbon from the atmosphere to one of the other 6 sink. So, one is the atmosphere, but the other 6 sinks are there where the carbon can be sequestered and possibly they can be converted to useful product also. Now, the point of controls are very important. Now, these are the 6 sinks that could be potentially utilized, except the 7th one, that is the atmospheric one, because we want carbon to move from atmosphere to the rest of the 6 sinks. Now, these 6 main sinks where the carbon pulled out, atmosphere, stored, and sometimes it can be utilized to produce different bio based products. So, that is the second interesting aspect of the entire story of the environmental biotechnology based carbon sequestration, because there we think of the circular economy or maybe a value chain system, that we convert the carbon, not only remove the carbon, but also try to convert the carbon into some valuable products. And the 3 biospheres. So, these 6 sinks together we can actually converge, we can think of converging them into 3 biospheres, which are considered to be the main targets for carbon sequestration to mitigate the global warming. And these are the marine system, that is oceanic, both the surface ocean and the deep ocean, deep subsurface of the terrestrial crust or the earth crust, plant soil ecosystem, which is where the entire soil is found to be the major part of the biome, where the carbon can be sequestered and can be utilized or can be converted to some form. Now, let us see these parts in little more details. Now, the deep subsurface particularly is less known, but in recent past, as I mentioned, last 2 decades we see very interesting observations are coming, and people are gaining interest in this particular domain of the earth, that this is considered to be one of the largest human caused releases of carbon into the atmosphere due to the extraction of fossil fuel. So, we have been continuously dragging the fossil fuels from this deep subsurface, but how about returning this carbon dioxide back to this deep subsurface? Because naturally, if we think back, the coal, the fossil fuel, the natural gases, these are the source of all the carbons that we are using today, and most of the carbon dioxide present in the atmosphere is actually coming from that, particularly the anthropogenic release component. But how about these that developing technologies, particularly biotechnologies, where the microbial functional capacity can be enhanced, so that the turnover of the deep carbon can be reversed, so that the carbon can be stored instead of carbon is lost as a gaseous form. And it is interesting that this microbial functional capacity within the deep subsurface of the earth crust remain largely unknown, and it has shown great potential for influencing the geological carbon storage. The second aspect is the plant soil ecosystem, or we conventionally call the rhizospheric region, because the primary producer are the plants who are naturally able to capture the atmospheric carbon dioxide and during their photosynthetic process they convert this carbon dioxide into complex organic molecule, and part of these molecules are being continuously pumped into the soil system, because the plants are having profuse root system and through the roots they release a large amount of carbon. Now, carbon can be stored in soil depending on the root system and how it interacts with the rhizosphere, that the root associated soil zone and the soil microbiome. So, there are actually three components. One is the plant root system, another is the plant root system associated soil system, and then the rhizospheric and the soil bulk soil microbiome, which are actually responsible for controlling the fate of the organic matter which is being released or produced by these green plants within the soil. So, these needs to be looked into in detail. Now, with respect to the engineering principle from ecological processes, the biotechnological solutions for CCSU will require the joint effort from multiple disciplines, including the chemical engineers, microbiologists, geologists, ecologists, plant scientists, and also economists. And the green shift concept has emerged, which is basically green shift we call towards technology built for sustainability, that has shown the potential to be the next big economic boom, and many political business agendas that support research and implementation of this plan stand to benefit by being early players in the movement. Now, first we will see the plants and soil. Soils are the foundation, as I mentioned, for almost all biological processes on the earth's land surface, and root soil rhizosphere function as a kind of carbon pump by pulling the atmospheric carbon dioxide captured by the plants and then below the ground transfer it and stored in the rhizospheric region. And in this region the plants interact with soil bacteria and fungi via the exchange of SOC, that is the soil organic carbon, that can be stored or respired back into the atmosphere. So, basically the root system is delivering the carbon which is originally fixed from the atmospheric carbon, and then they are releasing the SOCs. These SOCs are being metabolized by the organisms present in the rhizosphere in the bulk soil, and this SOC organic carbon, soil organic carbon, is getting converted into carbon dioxide, and the carbon dioxide or methane it is being coming out into the atmosphere. Now, the balance for this, that how much carbon will be assimilated by the plant and released into the soil as SOC, and then how much SOC will be oxidized by the soil microorganisms and they will be converting them into gaseous form. So, there must be a balance, and this balance for the adequate carbon sequestration is governed by the tight ecological interactions and localized geochemical factors. The engineering of the crop root systems for enhanced carbon sequestration promise basically to be feasible and to encompass mostly known risk. Now, in case of plant system, if we see, there are actually two aspects. So, one aspect is the plants can be selected to design having a very deep root system, creating the better carbon sink potential in the localized soil. So, the deeper the root, more carbon will be sequestered in the soil zone, because the rate of soil organic matter decomposition decreases with depth. Deeper it will be, less will be the oxygen level available, and the activity of the aerobic heterotrophic microorganisms oxidizing the organic carbon will be less. Plants can also be selected or designed to produce biopolymers that resist decomposition. For example, the suberin type of molecules, which are represented here as the black dots, which is a major component of cork and able to resist decomposition. So, plants can be selected who are able to naturally produce more suberin type of molecules, and these molecules will stop the ready decomposition of this organic material. That would possibly present an opportunity for crop based CCS. So, on the above ground we can grow the crop, harvest the fruits, vegetables, flowers, leaves, etc., but at the same time we can allow more carbon to be sequestered safely within the soil. Second is the deep biosphere. The deep subsurface is largely unexplored and remains a mysterious environment, and when we say it is a deep subsurface, it is few meters below the top surface, and it is referring to the biomes below soil, ocean floor, sediment, where community composition, microbial community composition and the functions, microbial community functions, are found to be different from those inhabiting the overlying organic soil layer or sediment interfacing the water columns in place of the oceanic system. So, the both oceanic deep subsurface and the terrestrial deep subsurfaces are found to play very important role in sequestering the carbon. Complex microbial communities are found to dominate these environments, both the oceanic crust as well as the terrestrial crust, and these organisms, as I mentioned, are not very well characterized, but they show potential high functional capacity for the turnover of carbon dioxide and methane. Deep subsurface is spatially heterogeneous from place to place because of the composition of the rock crust, and microbial functions also will change depending upon the depth, depending upon the location, availability of different substrate, etc., which also affects long term geological storage of the carbon. One of the approaches was the injection of the carbon into basaltic crust, where you can see that there are several efforts are being internationally done, where we can see that the water and carbon dioxide is injected into the basaltic rock crust, so that long term storage of carbonate minerals can be achieved. Now, this is found to be an attractive strategy. It is called geosequestration of carbon dioxide, particularly in the basaltic rocks, because the technology for injecting something into the underground system is already existed because of the microbially enhanced oil recovery or something similar, and it has already been applied for example, the oil recovery. Although geologic CO2 sequestration has a key role to play in managing atmospheric carbon dioxide, and already, as I mentioned, being practiced in several countries and several places, many current technologies are viable only for temporary storage, meaning that more innovation is required to fully understand the carbon dioxide sequestration and long term storage of the carbon, particularly considering the leakage factor. Hence, while the deep subsurface represent a major sink for CO2, more knowledge need to be gained, or we should understand how resident microorganisms present in these geological strata where the carbon dioxide is stored, so that we can enhance or limit the long term storage. Maybe the leakage can be controlled, or the enhanced CO2 carbonate formation or mineralization can be performed. Now, microbial biotechnologists have been developed to prevent CO2 leakage, from example from the wells and adjacent geologic formation in the deep subsurface. These innovations harness naturally occurring microbes, microbial process associated with the attachment, biomineralization and formation of biofilm underground, where the CO2 is being injected. The naturally occurring process will precipitate CO2 into the carbonate or carbonated sediment over a very long period of time, tens or thousands of years, demonstrating deep subsurface sequestration potential. However, innovations in microbial biotechnology has to be done, more has to be done, because we have seen that it is possible to enhance the carbonate precipitation and biomineralization, but more research and development of technologies are required. So, some of the aspects like the CCS biofilm technology, which help in 3 steps, like the microbial biofilm can actually allow in effective plugging of the pore channels over there, and we can see that there are definite advantages for this. Now, deep subsurface contains high microbial functional capacity for cycling carbon compound, and biotechnologies that seal the wellbore to reduce the leaking of CO2 or methane are being developed to manage the carbon storage. Now, there are 3 steps into this. These 3 steps are used to biocement the seal cracks, leaks or channels in the deep subsurface, specifically of the wellbore. Step 1 in this case represent the crack in the wellbore to seal. So, the microbes can form biofilm and help in sealing this wellbore, crack in the wellbore. The second step is injection or stimulation of the natural microbial community that form biofilm and produce an extracellular polymeric substance, EPS material, which will attach and stimulate growth along the cracks within the geologic formation. So, that is the step 2, and in step 3 bacterial biomineralization process can be induced, specifically the calcium carbonate precipitation that can act as a biocement sealant to seal the leakage, thereby allowing the long term sequestration and inhibiting the leakage of the carbon dioxide from the underground sequestering strata. Similarly, the marine microbiomes and microbial biotechnology are also gaining importance, because the photosynthetic marine microbes, specifically the algae and cyanobacteria, they have shown enormous potential. Marine phytoplankton, benthic microalgae, diatoms, they are all showing huge potential, because they can contribute to the 20% of the global primary productivity, for example. And despite the favorable biomass productivities, it is still widely understood that economic feasibility limits the application of industrial scale algal CCSU. Yet new value chain and socio-economic drivers for these technologies are emerging, which includes the food, feed and sustainable bioproducts, particularly from the marine organisms. And multiple industrial and pilot scale efforts are in operation, particularly with respect to the marine carbon sequestration and utilization, including Europe, North America and Asia. Now, marine microbial ecology offers actually multiple roadmaps for new CCSU technology, because our oceans play critical role in global carbon sequestration. Particularly, the marine microbiomes help facilitate to the photosynthetic primary productivity and resulting in large amount of dissolved organic matter or DOM being stored in our ocean. So, these can be harnessed, so that the more and more carbon dioxide can be converted into DOM by the microorganisms which are present in the ocean. It could be by the heterotrophic community member. The remaining fraction of the DOM accumulates, because heterotrophic community live in tight association with the algae and consume much of the DOM produced. The remaining fraction of the DOM basically accumulates and persist for several thousands of years. The molecular properties of this plankton derived DOM dictate the heterotrophic utilization and turnover rate, and it is found to be a global carbon sink and an ecological process that can be explicitly designed and engineered into algal mass cultivation effort to integrate the CCU, that is the biomass harvesting, to CCS, that is the marine storage of dissolved organic matter, and both the algae and other organisms can be utilized for that. Now, in the second part of this, we will talk briefly about the engineering the microorganisms specifically to enhance the CO2 sequestration, because we can understand that the microorganisms, whether they are in the soil, whether they are in the deep terrestrial crust or oceanic crust or in the marine marine environment, they play a huge role in sequestering the carbon dioxide. So, enhanced research interest or increased research interest has been shown to engineer the microorganism for enhanced CO2 sequestration. Now, because microbial CO2 sequestration is one part which not only provides the green and sustainable approach for mitigating this climate change challenge, but also it provides the opportunity to produce useful molecule like the biofuels and chemicals. However, it is facing some challenges, and these challenging factors are the efficiency of microbial CO2 fixation is found to be quite low, and concomitant microbial CO2 emission decreases the carbon yield of desired chemicals, and therefore, the strategies are identified to combat these issues. So, one aspect is engineering the CO2 fixation pathway, and energy harvesting system have been developed to improve the efficiency of CO2 fixation in both autotrophic and heterotrophic microorganisms. On the other hand, the metabolic pathways and energy metabolisms can be engineered, can be rewired to reduce the microbial CO2 emission and increasing the carbon yield of the value added products. Now, carbon sequestration from earth to cell is catalyzed by these microorganisms, where the microbes can actually remove the carbon from the atmosphere and can simultaneously convert it into the valuable useful chemicals, and some of them can be resources as good as to be used in bioenergy. Now, strategies for improving the microbial CO2 sequestration focus therefore on both the aspect. One is enhancing the microbial CO2 fixation, and second is the reducing CO2 release. So, we will look into these two aspects briefly. So, one aspect is that two methods are there and two strategies are there. So, what are these methods and strategies? Two methods might be able to, so one is reduce the emission, another is convert the carbon dioxide into valuable products. Now, two methods might be able to mitigate the global warming, developing CO2 capture technology and limiting CO2 release. And the strategies would be improving the microbial CO2 sequestration and enhancing the CO2 fixation and reducing the CO2 release. And as I mentioned, both autotrophic and heterotrophic microorganisms, they can fix carbon dioxide. Autotrophic microbes, they use the CO2 as the sole carbon source to directly produce the various value added chemicals. So, they convert CO2 into value added chemicals, including the molecules which are useful in biodiesel production, biofertilizer production, antioxidants and bioactive compounds. On the other hand, the titer and the rate of these chemicals which are produced out of the carbon dioxide, which is fixed by these autotrophic organisms, need to be improved. That is, for example, the highest rate of lipid production from microalgae is around 5 gram per meter square per day, while it is 30 gram per meter square per day is required to make commercialization of the algal lipid biofuel, which is considered to be economically feasible. On the other hand, the heterotrophic microorganisms, they can also generate chemicals with much higher productivity. For example, lactate ethanol and genetic tools for heterotrophic organisms are more advanced, including the CRISPR-Cas systems. Nevertheless, the CO2 fixation in heterotrophic organisms is based on using organic substrates to provide the energy, which actually leads to release of carbon dioxide, and net CO2 gain by microbes is achieved only when some specific products like malate or succinate are generated. Otherwise, there will be a substantial CO2 release. Now, the key challenge for CO2 fixation in case of heterotrophic organism is to provide an alternative energy source to the cells. Now, the reinforcing the CO2 fixation by autotrophic microorganism, as I mentioned, the CO2 fixation in autotrophic microbes can be simply described as a pathway which is driven by cellular energy, by like the ATP and the reducing power, and that transform non-biogenic carbon dioxide into some kind of a complex organic molecules through the CO2 or the bicarbonate which is taken in the cell is converted into acetyl CoA and pyruvate. Now, CO2 fixation can be reinforced by improving the efficiency of the CO2 fixation pathway. The way the CO2 fixation pathway operate, we can increase their efficiency by different engineering approaches, through developing more effective energy harvesting system that improves the supply of ATP and reducing power, and also regulating the distribution of carbon flux to optimize the utilization of cell resources. On the other hand, the reinforcement of the CO2 fixation in heterotrophic organism can also be done through carboxylation reaction in metabolic pathway, in which the substrate and energy for CO2 fixation both come from the decomposition of the organic compound. And the strategies for reinforcing the heterotrophic CO2 fixation may involve direct enhancement of native carboxylation, creation of an additional CO2 fixation bypass, and engineering the heterotrophic microorganisms which are capable of growing with CO2 as the sole carbon source. Now, these are the strategies. One strategy could be the native carboxylation, which can be enhanced by improving the activity of carboxylase and increasing the availability of the intracellular CO2. The other could be building CO2 fixing bypass efficiently, that will improve the heterotrophic CO2 fixation, and the other one is a fully artificial autotroph, may be constructed by building a complete CO2 fixation pathway and an energy harvesting system in a model heterotroph like E. coli by harnessing the power of modular optimization, adaptive evolution and other approaches of metabolic engineering and synthetic biology. The last one is the reducing CO2 release from the microorganisms. Now, during microbial metabolism, CO2 releases are there because of the decarboxylation reaction, because of the redox imbalance, and also because of the ATP generation to maintain the cell function. And there have been defined processes or strategies through which the microbial metabolism derived CO2, which is actually releasing CO2 into the atmosphere, can also be controlled, and these can be done using three categories. One is the rewiring of metabolic pathways, improving the redox balance and decreasing the respiratory ATP production. So, overall for these two aspects of the carbon sequestration, that the first one was a general aspect of the sequestration, capture, utilization, and the third one the more like how do we engineer the, or what are the options to engineer microorganisms for better CO2 fixation and reduced CO2 emission. These are the references, and in conclusion, the innovating carbon capture biotechnologies through ecosystem inspired solutions are discussed, and biotechnology and innovation efforts that are inspired and are connected directly to the three major biomes are introduced. It is important that biotechnologists and engineers seeking to build ecosystem inspired solutions will depend on fundamental expertise for a given ecosystem to understand which process, species, genetic parts can be rendered into controllable module, or perhaps more importantly those that cannot. The other message of the fundamental scientists is to help identify the value chain opportunities. How do we identify the routes through which the products can be synthesized, and gaining a deeper understanding of these processes, biologically catalyzed processes into carbon cycling will guide towards the innovation of industrial developments. It must be emphasized that better communication and cross disciplinary innovation will motivate future investments into science and industrial action to consider the benefit of ecosystem inspired CCSU. Microbial CO2 sequestration is a promising approach for closing the carbon cycle of Earth. Using current biotechnological strategies, the efficiency of CO2 fixation in autotrophic and heterotrophic organisms can be increased, and the problem of microbial CO2 release can be partly solved. Finally, the improvements of microbial CO2 sequestration often result in higher level of chemical production, which ameliorates the energy and resource shortage simultaneously. Thank you.